As Rakesh said, I'm going to be talking today about the work that I've been doing as part of my postdoctoral research fellowship here at the Hospital of Sick Kids in Toronto. And this work focuses on studying the structure of the V-ATPase within the native synaptic vesicle membrane. And so, as I'm sure you're all aware, the brain is a really important and complex organ. It's responsible for perceiving information, processing it, and then enacting responses to that. So whether that be motion or hormone secretion. And the brain on a cellular level is comprised of over 50 billion neurons, which relay signals from one to the other. The neurons in the brain interface to each other via synapses, laser pointer out, via synapses here. So each neuron can have up to around 7,000 synaptic connections, meaning there are estimated to be about 100 trillion synaptic connections in the brain, and these are all crucial for the brain activity. On a greater magnification at the synapse level, there's a process, there's a set of machinery that is highly conserved, highly regulated, which is involved in the loading, production of neurotransmitter, and then the release onto the postsynaptic neuron. So neurotransmitter is loaded into synaptic vesicles, and then these can be released as the signal enters the presynaptic neuron in order to transmit the signal into the postsynaptic neuron. And the V-ATPase is crucial in this process. So I'll just take a second to talk about this in a bit more detail. So within the synaptic vesicle, you have transporters that are important for the import of neurotransmitters into synaptic vesicles. And there's also a V-ATPase, which is a multi-subunit, very large, around 700 kilodalton proton pump that creates a proton gradient. And the V-ATPase takes ATP and hydrolyzes it into ADP in this soluble V1 region. And this hydrolysis caused the rotation of a ring within the membrane-embedded VO region. And this ring rotation allows the pumping of protons from the cytoplasm into the lumen of the synaptic vesicles to cause acidification. And this acidification by the V-ATPase is really important because these protons are then used by the neurotransmitter transporters to exchange in neurotransmitter into the synaptic vesicle. And in this way, the synaptic vesicles can be loaded. Can everybody see that? Or is it? I'm trying to hide it. I don't know. Never mind. So once the synaptic vesicles are loaded, they form the reserve pool. And at some stage, prior to docking of the synaptic vesicles, there's dissociation of the V1 region from the VO region. And this has to occur prior to vesicle fusion. So the vesicles will then dock at the presynaptic membrane. And when an action potential enters the neuron, so a nerve signal, there's an influx of calcium, and the neurotransmitters released when the vesicle undergoes exocytosis. And the neurotransmitter can then bind to receptors on the postsynaptic neuron, and this allows the signal then to be transferred into the postsynapse. And this cycle is then completed by recycling of synaptic vesicle components into new synaptic vesicles, reassociation of V1, and the cycle can then repeat as many times as needs to happen for the action potential for the neuron. And so basically, in my postdoc, the question I've been trying to look at is I've been wanting to see what the V-ATPase looks like within the native synaptic vesicle membrane. Because while the structure of the V-ATPase has been solved in a number of different environments, we were thinking it would be really interesting to see within this specific environment whether there are additional conformations or changes that rely on the native membrane. And so the first step of this was to be able to isolate the V-ATPase. And we did this from native source, so we homogenized a rat brain and then used our favorite target bait protein, SIDK. And SIDK is a protein produced by the bacteria that causes Legionnaire's disease, and it binds to host V-ATPase to help the bacteria evade the immune system. And we use this to isolate the V-ATPase on the synaptic vesicles. When we did negative stain of this, we could see the synaptic vesicles here. You can see like a clump here and a clump there, but they were swimming in a sea of free V1, so dissociated V1, which was making it really hard to be able to resolve anything about the V-ATPases that were actually bound to the synaptic vesicles. And so I introduced a synaptic size exclusion, step to resolve the V1 peak from the synaptic vesicles, and the resulting negative stain here you can see is much cleaner. You can see the individual synaptic vesicles with the V-ATPases poking out the side of them. So that was really nice to see. So the next question, once we had our synaptic vesicle sample, was to confirm whether they were truly synaptic vesicles. And to do this, we performed mass spec. And so this chart on the right, we have the input sample on the bottom, the number of mass spec counts of each protein, and we have the vesicle sample at the left-hand side, which is our sample. And essentially, we were looking for enrichment of key proteins, so moving towards this axis, to show that we had synaptic vesicles enriched over potentially isolation of other V-ATPase containing membranes. And so in green here, I've marked all the V-ATPase subunits that we were able to identify, and they're all really high abundance in the sample. And we were able to identify 14 of the 16 V-ATPase subunits, all bar these two small hydrophobic subunits, which were likely just unable to be detected via mass spectrometry due to their size. And then secondly, we also looked for common synaptic vesicle marker proteins. And so we saw a large number of these were really highly enriched in our sample, suggesting, well, proving that these are in fact bona fide synaptic vesicles. So we can see, for example, synaptobrevin and synaptotagmin here, and these are two snare proteins responsible for the fusion of the synaptic vesicles and the endocytosis. We can also see the V-GLUT neurotransmitter transporter that acts on glutamate neurotransmitter, which is the most common neurotransmitter, excitatory neurotransmitter within the brain. And we also see a large number of very common synaptic vesicle marker proteins that are enriched, including synaptophysin, synaptogyrin, etc. So once we know that we have bona fide synaptic vesicles, we wanted to see if they were active. And we did this using an activity assay, which incorporated a fluorophore, known as ACMA, that essentially is quenched upon the acidification of vesicles. And so prior to the addition of ATP, we have high fluorescence and a high pH, i.e. Neutral, inside the synaptic vesicle. And we saw when we added the ATP that there was a decrease in the fluorescence, indicating that there had been proton pumping in the vesicle, i.e. that our synaptic vesicles are active. When we added nigericin, which is a proton antiporter, and it essentially removes the proton gradient, we saw then a return to baseline. To confirm that this acidification and this activity was due to the V-ATPase, we replaced the nigericin with baflomycin, and baflomycin inhibits the V-ATPase, and you can see here we saw a slow leak, confirming that it was the V-ATPase causing acidification. And furthermore, if we incubated with baflomycin, so prior to adding the ATP, we saw that acidification of the synaptic vesicle even was completely lost. So now we know we have synaptic vesicles that are active. We wanted to follow up with some cryo work. And so I'm showing here a tomogram that was taken together, and this was work I did with Dr. Jijie Lee, who's our facility manager here at SickKids in Toronto. And you can see that we have a really nice distribution of the synaptic vesicles across our grid, and also of, you can see the V-ATPases in some of them. Slightly more clear to see in this micrograph. So instead of doing tomography, we actually did single particle analysis and collected a set of 18,000 micrographs, which I then sort of handpicked some of the V-ATPases and used model training to create a picker so that I could solve the structure of the V-ATPase within the synaptic vesicles. And so I was super lucky and super excited to see, after all the data processing, that it came out with this really nice structure of the V-ATPase within the synaptic vesicle membrane. So you can see here the vesicle bilayer with the V1 region sticking out, and the VO components are embedded within the membrane. And this resolution was about 3.4 angstroms on average overall. And it was good enough that we were able to build in a series of atomic models to make the V-ATPase structure. Really nice in there. One of the interesting things we saw in this structure was a series of densities around, so the ring of the VO region, this rotor region, that looked more or less the shape and size of cholesterol modifications. And this is really cool because this is one of the first times that cholesterol modification has been seen from native source. So it's been previously observed, but it's really nice to see it here because it confirms that this is a true density and not a contamination from a sterile-like detergent. And so that wasn't the only interesting extra thing we saw in this data set. We actually also identified an additional density, shown here in bright pink. And this density is bound to the VO region. It's relatively low resolution. So it's kind of high resolution here at the binding interface, and it gets further away the more you go into the membrane. But from this resolution, we were able to identify that it had 4 transmembrane helices. And looking at our mass spec, we identified 4 proteins that are all within one family that fit this, so synaptogyrin, synaptoporin, synaptophysin. And when we fitted these, so this is rigid body fit alpha fold models, just to be clear, to the density that we saw that the synaptogyrins lacked this luminal head domain that we saw in synaptoporin and synaptophysin. Looking then at the highest resolution portion of the map, so the bit at the binding interface, we could see 2 bulky side chain densities that were pretty well resolved right at the binding interface, which were fit really nicely by this tyrosine and phenylalanine on synaptophysin, but not so much by this alanine and threonine on synaptoporin, leading us to conclude that the protein that's bound at this density is synaptophysin. So moving on from this, once we had a really nice sort of resting state structure of the V-ATPase, we wanted to see if there were any conformational changes, for example, or if any of the things we'd observed changed upon loading of the synaptic vesicles. And so we treated synaptic vesicles with ATP and glutamate, the neurotransmitter, to mimic synaptic vesicle loading conditions before freezing to a grid. And for comparison for true unbiased comparison with the sort of resting state synaptic vesicles, a talented PhD student from our lab, Ryan Karimi, who's actually attending here today. So if you have any questions about this, please post in the chat for him. He developed a tool which basically identifies vesicles from your data set and in an unbiased manner picks particles around the edge of them. So it allowed us to do a really nice comparison of the resting state V-ATPases and the loading condition V-ATPases that we saw. And then once we collected these particles, we did pretty exhaustive 2D classification and came up with the 2D classes up top for the resting state V-ATPase, where you can see all of these have a V1 domain, VO domain, intact V-ATPases. And somewhat surprisingly, we observed that a subset of the 2D classes for the loading conditions data set lacked that V1 domain, suggesting that we'd observed dissociation of the V1 domain from the VO domain under these conditions. And so following this up, we wanted to confirm it via another method. And we also wanted to see whether it was the glutamate or the ATP or both that were required for this kind of fairly dramatic dissociation. And so we did this using a size exclusion assay. And so here, we incubated the synaptic vesicles overnight with either buffer, glutamate, ATP, or ATP and glutamate. And here I've got the glutamate and buffer curves that we obtained the next morning overlaid. And you can see that they overlay really well. So there's a negligible difference between the buffer and glutamate curves. However, when I then add in the curves ATP and ATP versus glutamate, they both show a shift in this V1 peak over towards the right. And they also show a decrease in the height of the synaptic vesicle peak. We wanted then to confirm whether this change in the height of the synaptic vesicle peak was due to loss of V1 or what was responsible there. And so we did a Western blot. And here you can see, so this is 3 repeats, and we plotted against SIDK, which is our bait protein that essentially acts as a marker for the V1 region because it's bound there. So we blotted against that, and we were able to see in the absence of ATP, there's pretty solid bands for the presence of SID-K, i.e. the presence of V1. And when we added in the ATP conditions, looking again at this one synaptic vesicle peak, we saw a distinct loss in the amount of SID-K present, suggesting that this was true V1 dissociation that we had observed on our synaptic vesicles. And so to summarize, we were able to get a high-resolution structure of the V-ATPase within the native synaptic vesicle membrane using single particle analysis cryo-EM. This data set really showed the importance of looking at proteins in their native membranes, as we could identify this synaptophysin novel interaction that we hadn't previously seen bound to VO, even in identical samples that have been solved from mammalian brain in detergent. And finally, we also were able to determine using this method some more information about how the dissociation of V1 happens from the V-ATPase. And so with that, I would just like to thank everybody here in my lab, in the Rubinstein lab, particularly Ryan, who was really helpful with the assays that I just showed you, and also the vesicle picker, as well as Dr. Giugilli, who helped with the cryo-EM data collection and tomogram collection. Thank you so much.
Thanks, Claire. The floor is open for the questions. Anybody?
Claire, I'm just wondering, how much do you think pH gradient and bilayer curvature drives the conformational change of the ATPase?
So I think, based on what I'm seeing from the ATP effect, so it's not glutamate, it seems to just be the ATP that's causing the effect, that it could well be the pH change that's causing the dissociation. It's being sent somehow through the V-ATPase, but I'm not sure if it's that or if it's just something to do with that actual hydrolysis of the ATP that's causing the dissociation. And then the curvature's actually a pretty interesting question. So synaptic vesicles are very uniform in size. They're all about 40 nanometers, 40 nanometers in diameter. And potentially we're starting to think about the role that membrane curvature could have within the V1 association, lesser the dissociation, as V1 association is promoted by a complex called RAVE, which appears to be able to bind best when the membranes are curved.
I think Barry has a question.
I'm sorry.
Yeah, Barry, feel free to unmic or unmute. Or I'll ask for you, unless Rakesh, do you want to ask Barry's question?
I can ask. You see no dimers. Is it possible that the additional synaptobrevin contacts compete for a dimeric interface that you would see, for instance, in the proton ATPase and the mitochondrial cristae?
I believe it might be ATP synthase in the mitochondrial cristae that's dimerizing. I might be wrong. But we don't see any dimerization, and we do see on these vesicles sometimes like 7, 8 V-ATPases per vesicle. But I don't believe that the synaptophysin biting has anything to do with dimerization. And indeed, a paper's been published also recently on a very similar piece of work, that they showed a structure of the V-ATPase without synaptophysin, so in a synaptophysin knockout. And they didn't observe any dimerization at all there either. So I don't think it's to do with the synaptophysin.
There is a question from Irwin asking, what type of size exclusion did you use for the synaptic vesicles?
Okay, so this was actually reasonably standard size exclusion. So it was a Super 6 10300 column on an Actopure, which worked really nicely for separating the V1 from the synaptic vesicles and gave us actually a really clean sample.
I'd like to invite anyone who hasn't asked a question before, and then Tim. Once Tim.
Tim has a question.
Okay. I'll go for it, Tim. But I really want to encourage people who haven't asked questions or who might be new to this meeting to feel free to pipe in with a question.
Yeah, absolutely. Yeah, so the cholesterol you saw decorating around the outside, do you have any thoughts on what that's doing? Is it just because it's obviously, if it's going to be bound, it must have a functional consequence. Do you think about what that was?
Yeah, so I'm not entirely sure. So we know that synaptic vesicles are pretty high in cholesterol, so like unusually high compared to other intracellular membranes. I think it's at about 40% cholesterol in the synaptic vesicle membrane. And currently, our best hypothesis is it's acting in a way to sort of lubricate the rotation of the rotor and sort of maybe shield it from the rest of the just sort of slightly more random assortment of lipids within the membrane. So to allow it to work a bit faster, maybe.
It does look like a lubricant when it's in there. So, but yeah, I mean, it'll be interesting. In some systems, you can actually swap out the lipids to a certain degree. So, for example, in the SMALp system, you can do lipid replacement and actually try and pull it out. Although I suspect if it's bound tightly, it's going to be very difficult to do that. So it's going to be a difficult one to remove, I guess.
Yes, Mike.
If no one else has a question, you guys have probably tried a variety of things. Can you mention what didn't work? Like, did you try different nanodisk systems or polymers, or did other polymers or nanodisk systems work for you? The native vesicles seem to be gaining traction and really amazing work.
Thank you. Well, yeah, so we wanted to do it fully native. So we didn't actually try any polymers. So the structure of V-ATPase was previously solved from brain. So very similar source organism, well, the same source organism. It was previously solved, but in detergent. And we just wanted to see really the importance of looking at things in native membrane by doing it fully native.
Mm-hmm.
So we actually didn't spend too much time trying different things, really. We just went straight for native.
Okay. And great paper. Where is it sitting now? Is it coming out soon?
Thank you. It's coming out on Thursday. So
Oh, great.
keep your eyes peeled.
Congratulations.
And any advice on for people here about getting published in this field where there's all these new advances and it's hard to get reviewers to understand maybe the latest technology?
Oh, that's a hard question. I don't know. I think always tying it back maybe to the basic biology and always highlighting the importance of the individual things and sort of giving big picture ideas instead of maybe getting more bogged down with the smaller parts that I enjoy, but I don't think the reviewers do. Might help, I think.
Okay. Well, great. Amazing story. And Rakesh, thank you for